Okay, so want to use our time efficiently. Normally, what I'd like to do at the beginning of this is to just kind of go back, explain where we are, how we got here before starting this. Really, the end of the line, so to speak, for glucose metabolism, just kind of a summary of what it is that got us here. But as you know, we're kind of pressed for time. I don't have a lot of time to do that, to go through the whole pathway again. So, yeah, just look at me. We're here. So continuing onward from here, we're going to talk about what today really is the end of the line for glucose metabolism. In our next lecture after this, we'll start to bring in other molecules. We've already done a bit on amino acid oxidation, so we're going to get into that. Really focus, therefore, most of the rest of the time on getting lipids both metabolized and then also looking at how they're synthesized. But today we're going to be talking about this really cool system called the electron transport complex, oxidative phosphorylation, and chemiosmosis, which again, as far as we were talking about glucose metabolism, really is the end. This is where we're actually now going to make most of that ATP that you have to use. So really, to summarize a little bit of where we are, we talked a lot, especially in Biochem 1, about this idea of having potential energy, and the idea that energy is really whatever it is that makes something possible, makes something happen. But more significantly, the idea that if we have some energy, some stored potential energy, that we can couple to that potential energy as it is used to do something productive. In other words, to couple endothermic processes, exergonic processes to endergonic processes. And most of what we've seen so far has been chemical potential energy. That has been driving reactions forward. We're dealing now with a situation where, as a consequence of all of this, and I'm not just bragging about this, as a consequence of all this, we have an extraordinary amount of electrical potential energy, which is very, very different from any sort of potential energy we had before. Now it's not so much the numbers of molecules we have, but rather those molecules being in a highly reduced state. And we'd like to do something with all that energy. And so to do something with all that energy, we have to find ourselves in a situation where we can position some molecule to accept those electrons. The molecule that we're going to position to accept those electrons in this case is going to be oxygen. The best thing of oxidizing it all. There's a reason why it is at the top of that table of reduction potentials. Nothing, at least in biochemistry, is really as good as the simple electron. And so we can do a calculation similar to the one we did last semester, where we could say just how much energy would there be if we were to take a mole of this and ADH, fully reduced, which is where most of our electrons are presently stored, and just allow those electrons to be transferred onto oxygen. Here are the potentials. Here's the differences under standard conditions anyway. Quite a lot. That's a volt. That's a battery. That is a volt. Quite a potential difference. And that's equivalent to 230 kilojoules per volt. So even under standard conditions and non-standard conditions, that is a lot of biological energy. That is a lot of energy capacity. And remember, of course, we have multiples of these NADHs that have been extracted through glycolysis, pyruvate dehydrogenase complex. We have another electron acceptor as well, FADH2, which we can also transfer those electrons on. That's what we're going to try to do. We're going to try to allow those electrons to move and to do some coupling. The key, and this is really the key on nature's part, is that we're not going to try to do this in one big step. Every time we try to transfer a large sum of energy in just one big step, a sufficient or significant amount of that energy gets lost. Right? When we light a fire, we do combustion. That's the same sorts of chemistry as breaking down glucose and sugar molecules, carbohydrates. But of course, most of that gets lost in the form of heat and light. And that's not what we want to do. So instead of just transferring all this energy in one gigantic step, we'll see that we're going to transfer in a series of steps, and that's going to allow us to maximize our utility of the energy, make the system more efficient. So far, we have made some ATP by a process that is known as substrate-level phosphorylation. We actually did that both in glycolysis. We also did that in the citric acid cycle. Even though the enzymes were the same and the pathways were slightly different in terms of what enzyme was handling which, the process was effectively the same. We made a very, very high phosphotransfer potential carbon molecule. We did so, remember, by transferring an inorganic phosphate to some sort of thioester. That was critical there in both glycolysis and the citric acid cycle. And And then we had enzymes allowing the phosphate to hop down its phosphorylate transfer potential. That's all the ATP we've made so far. That has been a process known as substrate level phosphorylation. And it's okay, right? I mean, for glucose, we picked up 4 nucleotide triphosphates. So that is not a bad number of nucleotide triphosphates. But it pales by the amount of energy that we still have available. And so we'll see that moving forward, we are not going to be creating high phosphorylate transfer potential phosphate groups and allowing them to hop down phosphorylate transfer potential molecules. We're going to use a different process entirely. This is what we've done so far, but this is a process that generates a bit of ATP, or say more generally nucleotide triphosphate energy, but it's not good for generating massive amounts of nucleotide triphosphate. So we're going to extract that energy in a different pathway. Just as a quick reminder, a couple of important biochemistry 1 concepts that we'll need. We talked last semester about the idea of transporters. These are proteins, membrane proteins that allow selectively some molecules to pass through the membrane. We talked about the idea that one could have uniport, full of active and passive. That's a single molecule that's capable of going through. We also saw symport. That's when 2 molecules need to pass through the membrane together for effective transfer to happen. And of course, antiport, where they need to move on opposite sides of the membrane. So we're going to see there's going to be some important membrane proteins here that are going to function as different types of transporters. Remember, again, we talked last semester, just briefly, when we were about membrane proteins, about the idea that if you do have active transport taking place, that can either be primary, where the energy is directly used to move a molecule opposed to its concentration gradient. There's a mechanism to this. Phosphate group with phosphorylated protein changing its conformation, allowing you to take up one of these X's and then change the conformation again, allowing it to be released on this side. But we also had secondary active transport, in which case we use a primary mode of transport directly, using again ATP to cause those conformational changes, establish a gradient, and then allow that gradient in turn to move another molecule across its gradient. And so this is what we call secondary active transport. This is just kind of a brief review from Biochem 1 because it has been a while. Okay, so again, where are we? We are in the mitochondria. Citric acid cycle has all taken place here, almost all of it in this soluble, this gluey matrix material in between the double membrane. At least one of those proteins has actually taken place in the membrane. Just as a reminder about the mitochondria, it is this double membrane organelle. The outer membrane here is permeable to many things, lots of things, such that the space between these 2 membranes actually has a composition that's fairly close to that of the cytosol. So we actually call this the C side. Again, it's not the cytosol, right? There is a membrane separating this out from the cytosol, but its composition is kind of close enough that we can call it the cytoplasmic or cytosolic side. And then we have the second inner membrane here, this one that is heavily folded over, has an extremely high protein concentration, a very, very large membrane, and this membrane is almost permeable to nothing, right? This is a real barrier, this is a real fence, such that the composition of the matrix is very, very different than that of the outer membrane space, or even that of the cytosol. Here is your typical beam picture of the mitochondria. Here's an electron microscope image. We've talked about the mitochondria a bit, but some of this will be actually relevant for our discussion. So if we just go a little bit further into the mitochondria, the first thing you need to know is that picture of the beam is completely wrong. I mean, it's the one that's probably still in your textbook, unless you just bought the most recent version. But this image, or this idea that the mitochondria are these localized, discrete, bean-shaped structures that are just kind of floating around the cytosol is not correct. What you're actually seeing here is an image of a cell, and all of this red is actually a fluorescent marker for where the mitochondria are. And what we can see is that rather than seeing the mitochondria as being these little sort of beans individually floating around, the mitochondria are actually dynamic organelles. They are constantly coming together, fusing and breaking apart, exchanging components with one another. This is really critical to maintaining the correct health and the composition of the mitochondria. This bean-like picture that you have was because this was kind of the best images that one could get for a long time. I mean, this is what you do see if you look at an electron microscope. But in fact, the mitochondria are not just these little static beams with heavily folded membranes inside. They are a large contiguous structure that spreads throughout a good portion of the cell. And they're very dynamic. They're coming together, they're fusing, they're breaking apart. That's really quite significant, because that's necessary to ensure that mitochondria always have the right components to be carrying out the functions that we'll be describing for them. The other thing that's kind of really cool, I mean, I always go back to these because I kind of love these slides. We don't need to work through the whole math, but it's right here. But again, we talked about the fact that this inner mitochondrial membrane is very heavily folded. If you were to unfold your inner mitochondrial membrane that is present inside of you, all of the mitochondria inside of you, if you were to unfold these heavily invaginated membranes, you'd have 26 football fields wide of mitochondria. So that's how intensely folded on top of each other they actually are. 26 football fields worth of mitochondria are packed inside of your body. So, I mean, I think that's kind of interesting as well. Anyway, all of this taking place in the mitochondria. That's where we've been for a while. Mostly soluble proteins, excluding that succinate dehydrogenase. Let's return here to the chemistry. So this was the calculation we did before. Most of our electrons are in the form of NADH. This is a pretty good calculation to do. You can see that there's a pretty steep potential there. You can get quite a lot of energy out of that. But again, to reiterate the point made earlier, not a great idea to try to dump all that energy or all those electrons directly onto oxygen. That would be a very efficient process, and so we're going to do so stepwise. And so we're going to do so by transferring those electrons gradually down what we call a series of complexes. These complexes are membrane protein complexes that have cofactors associated with them that can accept the electrons, and they're going to gradually move down their electrical potential and in turn convert some of that energy into a form that can be used to make ATP. And so you can see that these complexes here are lined up in terms of their relative free energies that are calculated. We start with our NADHs or FADH2s, and we're going to gradually move these electrons from complex to complex. Eventually they will come to rest on an oxygen. That is the most oxidizing thing. And when they pick up some protons along with that reduced oxygen, we'll have, of course, water, which is where pretty much all the water on Earth produced today now comes from. What are the electron carriers? Just to be clear, in general, most of these are associated part-time or full-time with the protein. So you see some proteins here, but you also just see the carriers. We'll kind of focus mostly on the molecules, but keep in mind that there are proteins that are involved and are associated with this, okay? So what are these various different types of electron carriers that we're going to see? One of them, kind of familiar, we've seen that one a lot, that's NAD, or it's produced more NADH. We also have a bunch of different flavins, sometimes connected to a sugar or another nucleotide here, sometimes just in this form. Remember the difference between the NAD and the FAD are twofold. One is that this guy can only take or release 2 electrons at a time. This one can do a 1 or a 2 electron reduction oxidation, so that makes it a bit more versatile for electron transport pathways. Of course, the NADH is capable of leaving and going between different enzymes. That's not true in the case of the flavins. They are tightly associated with the proteins that they exist with, and so if you need to move something between different proteins, the flavins are not going to be there, right? That does confer a certain advantage on the flavins that are missing from NAD, and that is that the proteins themselves can have a very, very big impact on what those oxidation reduction potentials are. In the case of the NAD, the protein binds so weakly to the NAD or the NADH, depending on what form it's in, they really don't affect the oxidation reduction potentials at all. They're from very, very loose association. You come by, you pick up some electrons, you go there, you drop off some electrons. The potentials are the potentials. But because of the tight associations between the flavins and their proteins, they can actually span quite a wide range of oxidation and reduction potentials. And so the proteins can actually tune them as to how good they are at accepting or releasing electrons. And that's actually quite important. And that's why it's significant that we have both ones that are permanently bound and also those that are capable of just floating between different proteins. Anyway, our next one that we should look at, something we talked about also briefly in Biochem 1, were the ubiquinones. Ubiquinones are kind of like a hybrid, if you could will, between the flavins and the NAD. They are like the flavins in that they can do 1 or 2 electron oxidations and reductions. So you can pick up 1 electron, or you can pick up 2 electrons by a ubiquinone. But they're like NAD in that they can float between proteins. And so where do we find this particular hybrid? It turns out that these are actually immersed, so they're part of the membrane. So these are actually physically completely enclosed in the membrane. These are not soluble at all. So it's not that there are like a compromise between NAD and FAD. This is when we need to move electrons between membrane proteins, 1 electron or 2 electrons. You can see Cytochrome C, there's the heme group there, that's where the electrons are being carried out. Again, those can carry only one electron, we'll see the consequences of that later. Iron sulfur proteins, we've seen them already when we talked about RNA, and we've also seen them when we talked about iconocase and citric acid cycle. Here they're doing their more conventional function, which again is to serve as batteries. So all the electron oxidations and reductions are happening here. They come in a number of different forms, so this isn't the same protein transforming between different structures. These are actually different structures, but depending on the arrangement of sulfurs around the iron, that gives them different voltages, again, for being oxidized or reduced. Anyway, that's our collection. That's the molecules that are going to be moving the electrons through the electron transport chain. So, we said that we're going to be moving a series of electrons through the electron transport chain from protein 1 to another protein complex, so on and so forth. Not a lot of time, again, to talk about data, some really interesting experiments. But we should at least discuss the question of, well, okay, so we have all these different proteins and these different cofactors. How do we know which order they go in, right? So I'm going to present to you a specific order, and I can't show you really interesting experiments. I can't show you all the really interesting experiments, but at least let's try to get some sense of how it is that we know what it is that I'm about to present here. So how do we know which cofactors the electrons hop first here, and then to there, and then to there, and so forth? There are really three different ways in which one can do so. One way one can do so is to just take those reduction potentials that you have in your fact sheet and line them up, right? You know, oxygen's up top, that's the most oxidizing, and then the one below that will give electrons to oxygen, and so on and so forth. And it turns out, remarkably, that if you do that, you actually can do pretty well. So in other words, just by measuring those potentials for the isolated protein components in the lab, you could come up with a pretty reasonable pathway by which electrons are going to move from cofactor to cofactor. It doesn't work so bad, especially for just, a table that you have in the fact sheet. But of course, we want to do this experimentally as well. We want to determine, okay, well, how do we know, right, that that is correct? How do we know that that's viable? And it turns out that there are really two experiments that one can do. But what I need to tell you about both of these experiments, so that it applies to them collectively, is that all of these electron acceptors, these molecules that are getting oxidized and then reduced in the pathway, absorb light differently if they are in the oxidized form or if they're in the reduced form. So they'll absorb light at one wavelength if they're oxidized, and they will absorb light at another wavelength if they are reduced. And so we can actually record a spectrum, right, of the electron transport complex, and that spectrum would look different if the molecules were oxidized form, if they're in the reduced form. So the first experiment, the first set of experiments that were actually done, were to actually take an electron transport complex, isolate some mitochondria, isolate the proteins that I'm talking about, isolate them in the absence of oxygen, and put some NADH in. And what's that going to do? That's going to result in all of the molecules of the electron transport complex having an electron on it, right? Whatever the particular order is, the NH electrons are going to go to whatever the first one is, and then they'll move to the second, third, fourth, and fifth. But then the next electron, the next NADHs, will kind of fill them all up. So we will have a completely full, completely reduced electron transport complex, right? Everybody has electrons on them now. It's like one subway stop gets jammed, and so the subway behind it gets jammed up, and the subway behind it gets jammed up. The whole subway system is now shut down. And then what one can do is to allow some oxygen back into the system. You allow oxygen back into the system. So what happens? The electron donor directly to oxygen will be the first one to go from the most reduced form to the most oxidized form. So you'll see it disappear in your wavelength, in your spectrum. And then, as that starts to happen, as the one that's immediately before oxygen starts to be depleted, the one after it will also start to be depleted as it starts to try to move its electrons over to the one that's immediately before oxygen. And so you can see that you can work backwards from oxygen, right? The first one to disappear will be the one that donates directly to oxygen, and then the next one will be the one before that, and then the last one to disappear should be the one that first accepts the electrons from the NADH. And so those sorts of experiments helped us establish it. Now, those are good experiments, but these things are happening really quite fast, and the spectrum aren't quite as clear nice if they were just clean, perfect spectrum. This one goes, and then this one goes, and then this one goes. But of course, this is a dynamic process. As soon as that first subway car goes moving, they're all moving. And so it's not like one disappears, then the next disappears, then the next disappears, and so on and so forth. So you would like to have some other way to validate that you got the order in which the electrons are moving is actually correct. And fortunately, you do. You have a series of molecules that are known from independent experiments to disrupt specific complexes. So for example, consider rhodinone. You could take your NADH, right, add it to the system. If you have rhodinone there, everybody else is going to stay in the fully oxidized state. That must mean that whoever's accepting the NADH, this complex here must be first. Now, suppose instead of putting rhodinone into your experiment, you put the antimyosin N. You put some electrons in, the NADH will get reduced, the quinone will get reduced, the cytochrome B will get reduced, but this process here is being blocked. So clearly the actinomyosin A is blocking this one right over here. And so by putting in these selective inhibitors, we can kind of trap the system. And again, the reason why we need to do that is because if we try to do this without these inhibitors being present, it can be very, very complicated to follow the kinetics of who's becoming oxidized, who's becoming reduced. These things are happening really fast. So these inhibitors allow us to trap the electron transport complex in discrete steps. Cyanide's a really great example, right? If you run this whole thing, add NADH and put in cyanide, all of these will become reduced, but the oxygen, right, will stay oxygen. It will never become water. That means the cyanide is blocking this process over here. And so through this combination of using the actual reduction potentials from the table, these experiments where you're allowing oxygen to sort of allow things in, to be watching who disappears first, and also using selective inhibitors to block different parts of the process and seeing who remains reduced and whose state becomes oxidized, that allows us to work out what the order is, and that order is actually here. It's what we call the electron transport chain or the electron transport complex. So look, I'm going to say protein a lot, but it is really, really critical to realize that none of these are isolated proteins. These are complexes of proteins. We're talking about 40, 50, even more than that, polypeptide chains and all of their associated cofactors. It's easier just to kind of call collectively the whole thing a protein, but when I say complex, I mean a complex of a lot of proteins. Anyway, here it is, and let's try to understand what's happening. Let's first try to understand the direction of the electron flow, and then we'll work through this step by step to see everything that's going on. We know we have a bunch of NADHs. These were generated by the citric acid cycle, not, by the way, by glycolysis. Only by the citric acid cycle. It's going to drop off as electrons here directly, and yeah. Those electrons from the NADH are going to be transferred to a molecule called quinone, Q, which is pre-existing in the oxidized form and turned into QH2. Okay, so the Q is going to come in, take those electrons, become QH2. That's complex 1. And then we go to complex 3. And I know you're thinking, wait, whoa. You skipped 2, Dr. Astra. I didn't skip 2. They just put 2 in. It was discovered when all of these various proteins were being characterized. It's kind of hanging around, but if you think of like complex 1, 3, and 4 as your friends, because those are actually involved in the process, complex 2 is like that person who's always hanging around your friend group, right? They were there, someone might mistake you for being one of your friends, but actually complex 2 is not part of the electron transport complex. It is going to play a role, but strictly speaking, it's not really a complex. So first, a little bit of confusion out of the way. Anyway, so the electrons go from 1 to 3, riding on the quinone. In 3, they will get transferred to 3, so the quinone will get unloaded in 3. One at a time, those electrons will get loaded onto a protein called cytochrome C. Cytochrome C is, strictly speaking, a soluble protein, but it has such high affinity for the membrane, it kind of hangs around here. Cytochrome C will bring these electrons over to complex 4, where they'll be transferred down to oxygen to make water. That's the electron transport complex. 2 open-ended questions then. First, what's going on then with complex 2, since complex 2 is not really a complex? Complex 2 is actually just that enzyme that we saw in the citric acid cycle. You remember that the succinate got converted into the fumarate? That's what complex 2 is. The membrane protein from the citric acid cycle. It does have another important role. You remember we also left those electrons on the FADH2. We know electrons cannot leave FADH2 and just go to another protein. So the other role of complex 2, and we're going to call it that because that's what it's technically called, even though it's not really a part of the chain, is going to be to transfer them from the FADH2 to the quinone. So our route is going to be 1 to 3 to 4 for NADHs, or for those that got stripped off in this step here where succinate got turned into fumarate, 2 to 3 to 4. Important implications for that difference. Others the question, why are we even doing this? Like, what's the point, right? We're just shuttling electrons from one place to one place to one place. And the key to understanding that is to remember that we are always moving electrons down their potential, right? So the electrons are starting off here at a very high potential. We know we're moving them to a lower potential, which means energy is going to be released. The question is, what are we going to do with that energy? And the answer is, not make ATP now. We're a little bit away off from that. Instead, we need to have an adapter. We need to put that energy into a form that can be used by an enzyme that synthesizes ATP, and that energy is going to turn out to be a proton gradient. So every time the electrons are moving through these complexes, complex 1, 3, and 4, remember I said 2 is not really a complex, 1, 3, and 4, some electrons are going to be pumped, taken, scooped up from this side of the membrane and pushed onto the other side of the membrane. That's what the energy is going to be used for. Conformational changes will take place. Every time an electron hops onto a new molecule, will change the structure of the protein in such a way that it can gather protons from this side of the membrane and move them to the other. How many? 4 in complex 1, 4 in complex 2, and 4 in complex 3, and 2 in complex 4. What about complex 2? Again, it's not really a complex. It doesn't pump any protons at all. It was just kind of discovered at the same time. And if it seems like I'm being kind of weird about that nomenclature, I will show you that that's actually is experimentally a very good way to describe things. And that is because if you look in the membrane, despite my picture that I showed you here with 1, 2, 3, and 4 all lined up, these complexes are not spread out over the cell membrane. Complex 1, 3, and 4 are actually in these structures that are known as restrosomes. They are sitting right on top of each other, packed together. And what is actually holding them together, complex 1, 3, and 4, is this lipid here called cardiolipid. We talked about this a bit last semester. Makes a major component of the inner membrane, mitochondrial membrane lipids. It's actually acting like a belt, holding all of these complexes together in one place. Not complex 2. Complex 2 is in the same membrane, so it's not a surprise that it's found together, kind of floating around there, but it's not directly associated with complex 1, 3, and 4. And if you take the cardiolipid out of the membrane, which you can do, this still works, but it becomes very, very inefficient. You produce much, much less energy, much less ATP in the absence of cardiolipid. So it's not that these things coincidentally happen to be found together. It only works efficiently if they're found in these structures called restrosomes. Anyway, enough said. Let's take a look at these complexes. Let's actually see what's happening. Let's try to see how we're going to generate that energy into a form that an enzyme can actually use to make ATP. So here we go. Here are NADHs from the citric acid cycle, not from glycolysis. That will turn out to be very important. We're going to drop them off with the first electron acceptor, FMN, and it's going to shuttle those electrons from one iron sulfur cluster to the next iron sulfur cluster. Every time you do one of these transferings down the well, the protein changes its shape to the extent that it undergoes a conformational chain that moves 4 protons from this matrix side to the intermembrane space. Okay, so that's what this complex of 46 proteins and iron sulfur clusters is going to do. Extract the electrons from the NADH and take 4 protons from this side to that side. Now we are later on going to count the number of protons that are actually moving, but what's going to make this a little bit tricky, and we will see the simplification, is that 4 protons are being pumped. So you're moving 4 protons from this side of the membrane to this side of the membrane, but that's not the imbalance in protons you're creating. What do I mean by that's not the imbalance of protons you're creating? Well, at the end, this iron sulfur cluster is going to transfer those electrons on Q to make QH2. So those 2 electrons will get transferred to the quinone. QH2. So even though we're only pumping 4 protons across, we're taking 2 more out of the matrix. These aren't getting pumped. These are being used to make QH2, right? Okay, so we're actually depleting by 6. But we're actually only depleting by 5, because of course we're dropping 1 off when we make NADH. So you can see that the number of protons that's pumped is not the same as the number of protons that are being depleted at each step. At least not now. The stoichiometry will all add up in the end, but it is a bit different. Anyway, here's what's going on in complex 1. If we could just kind of zoom in on the quinone site. We start with a fully oxidized quinone that is present in the membrane. There is fully oxidized quinone. It comes in, it picks up 1 electron, it picks up 2 electrons, and then it leaves. So you have the fully oxidized quinone and then the fully reduced quinone. There is no semiquinone in the membrane. It binds too tightly to these various proteins. So the only thing you'll ever find in the mitochondrial membrane is a lot of this fully oxidized quinone and some of this fully reduced quinone, and we just saw the first place in which we generated. Here again is complex 1. People are still trying to work out what the mechanism of this is, but it seems again that there are these alpha helices here that actually are acting like pistons or pumps. Every time the electrons move, it pushes one of these pistons down into the mitochondria, and every time it hops to the next one, they shift back up. So it looks like these alpha helices are literally binding protons and then just pulling them to the other side of the membrane. Here's the crystal structure of the entire complex. The mechanism by which this works is really still not understood, but it clearly involves some motions of these sliding backward piston-like motions of these various alpha helices, pulling protons from one side of the membrane and pushing it to the next. Anyway, the quote-unquote complex 2, not pumping any protons, it's really just part of the citric acid cycle, but it does have the other job, in addition to just doing that succinate fumarate oxidation, it also transfers them to QH2 as well. So again, you have a lot of quinone in the membrane, we've generated some QH2, the reduced quinone in complex 1, we have generated some reduced quinone in complex 2. The difference is that this is not a proton pump, right? It is just an enzyme we've seen before in the citric acid cycle. So it's here, it was found hanging around, named it complex 2. It is not in the electron transport chain directly. It's like a side branch, if you will. Anyway, now we have to get the electrons forward, right? We need to get them off these quinones, and we need to get them further, and of course, in doing so, we're going to be moving electrons down electron potential paths. So ideally, we can do so in a way that we could pump some more protons, create more of a gradient on opposite sides of the membrane. So how are we going to do that? That is the job of complex 3. Let's remind ourselves what we have now. We have a collection of quinones that are known as the Q pool. The Q pool is a mixture of mostly fully oxidized quinone, Q, just floating around the membrane, not associated really with any protein, and fully reduced quinone. And we know we have on each QH2 2 electrons, but unfortunately, the protein that's going to move us to complex 4, cytochrome C, only takes 1. If there's any evidence that nobody designed this, it's that the protein that's going to move us forward can only accept half of the number of electrons at a time. So if you are confused by the process that I'm about to tell you, thinking, no, wait, I'm lost, that's not right. No, you're correct. You really are confused. This is a very confusing way to kind of connect, a square to a circle. You don't try to adapt it, right? This is what nature came up with to get us to complex 4. We have the Q pool. We have complex 3. Our goal in complex 3, of course, is going to be to pump protons. We're actually going to pump 4, so we know that. But also to move those electrons forward. The QH2 has 2. Cytochrome C can accept 1. So we need to have a bit of an adapter here, if you will. Complex 3, here's our adapter. There are 2 different places, 2 different binding sites for quinones in complex 3. But these binding sites are not equivalent. One of them will only accept a fully reduced quinone. The other can only accept a fully oxidized quinone. One will only accept a reduced quinone, one will only accept a fully oxidized quinone. What happens? Each of those bind into their respective binding sites. A cytochrome C, which can accept 1 electron, binds at side of this protein. One electron goes up from the QH2 to reduce the cytochrome C, and it leaves. It's done all it can do here. The other electron travels downward to take the fully oxidized quinone and generate the semiquinone. This quinone here leaves complex 3. It doesn't fit. Remember, that binding site can only fit a QH2. It does not fit a quinone. So it leaves. This semiquinone is trapped. It cannot leave. It binds just as well to that site as the fully oxidized quinone. It's just that there's no semiquinones floating around the membrane. 2 protons get pumped. Now another QH2 then comes in and binds in this first binding site. It slides in, binds in that binding site. Another cytochrome C comes in. This is a different cytochrome C, right? One of that other cytochrome C is long since out of here. And the process repeats again. 1 electron goes up. 1 electron goes down. QH2 has lost 2 electrons, as well as these protons, which become pumped. This does not fit, so it returns to the Q pool. But who else returns to the Q pool? The second quinone in the form of QH2. Right? So we had to essentially move a pair of electrons to this quinone just to send it back to where it started. And so when I say that this looks like a little bit of efficient, notice what we had to do. We needed 4 electron transfers to move 2 forward, and 2 ended up just back where they started again. This is because the cytochrome Cs can only take a single electron at a time, right? So we generated the quinone, it left. We put another quinone, it left. Those electrons were moved forward, a pair of them, but the other half of them were used to reduce this fully oxidized quinone into a fully reduced quinone, which then went back into the Q pool, only to repeat the process again. So it took multiple electrons to move those forward, but I mean, anyway, that's okay, because each time we were able to pump 2 protons, so we pumped another proton across the membrane, so now we've pumped 8 protons. All right, finally we reach our last complex. Complex 4, also a proton pump, so that's beneficial. This is going to happen in 2 discrete stages, right? Because remember, we have 2 cytochrome Cs coming in, so that's going to be really critical. Also critical are going to be the specific electron acceptors, the various cofactors that are actually present. Most of the interesting things are going to happen here at this iron-copper combination. What happens is the first 2 cytochromes come in, 1 and then 2, and they're going to reduce our iron 3 to iron 2, copper 2 to copper 1. So they're going to dump those electrons off. First two come in. Okay? Now, an oxygen can bind to this state and form this peroxo bridge, and it's going to suck those electrons basically right back out, because that's what oxygen does, right? It's going to suck those electrons back out and return them to their default state. Another 2 cytochromes are going to come in, along with a pair of chemical protons. These are different than the pump protons. Remember, chemical protons pick up on molecules. Pump protons move to the opposite side of the membrane, generate the peroxos, and with another 2 protons, we just generated a pair of water molecules. In this whole cycle, a total of 2 protons also get pumped. So we generated 2 water molecules, we pumped 2 protons, we've depleted a bunch of protons, but this is the end, right? All those electrons that used to be present in glucose, the molecule that we actually started with, are now actually found in these molecules of water. So water is basically just our reduced oxygen. Keep in mind, and this is quite significant, that the stoichiometry of working itself through the electron transport complex just became very, very non-regular. Because a single NADH was not sufficient, right, to generate a pair of water molecules. You'll notice that it took 4 electrons or 2 NADHs to complete the entire process. So to complete the entire process, 20 protons had to be pumped, right? 4, 4, 2, 4, 4, 2. So when you're considering the number of protons that are being pumped from one side of the membrane to another, It really depends what you're defining it respect to. If you're defining respect to an